In the mid-2000s, I was part of a research team at the University of Utah, which collected and measured isotope ratios of tap water from across the United States. The goal was to develop a model that predicted the isotope ratios of tap water people were drinking in different parts of the country. When we first reviewed the data, it was obvious that it captured some clear spatial patterns. High and low values were each clustered in particular regions, or to put it in another way, cities that were closer together tended, on average, to have more similar tap water isotope values. This is an example of the phenomenon of spatial autocorrelation, which is widespread in natural and human systems. For example, in the density of tree cover in US cities, or the distribution of household income across the Salt Lake Valley. Spatial correlation is important in many kinds of spatial data analysis. Sometimes we can see it clearly with our eye, but we can also quantify spatial autocorrelation with statistics, such as Moran's I, which takes values ranging from -1 for data where closely spaced sites have very different values, to +1 when close sites are highly similar. In the case of our tap water isotope data, the relatively high value of Moran's I is consistent with our visual interpretation of strong spatial autocorrelation. This is good, because spatial autocorrelation provides a basis for spatial interpolation, the prediction of values at unsampled points based on their spatial location. If you have ever drawn contours on a map, you have implicitly done spatial interpolation. But contouring by hand means making subjective decisions about where to place the lines. We can do better. The Kriging method starts with the premise that the observed values represent a spatial process with a mean value over the area of interest and a spatially autocorrelated error term. Different Kriging methods treat the mean value as fixed or allow it to be modeled in different ways. But the crux of Kriging is to model the value of the error term at a given location as a spatially weighted average of error values at the observation sites. To do so, we need to estimate the weights, which will reflect the stronger relatedness of values at more closely spaced sites. This sample semivariogram shows this relatedness for all sample pairs in our tap water dataset as a function of the distance between sites. Semivariogram plots are inherently noisy, but for data with good spatial autocorrelation, the semivariance values should be low for closely spaced points and rise towards a plateau at larger distances. To fit a model, we usually average the data in bins that represent even distance intervals, and then fit a parametric model to the binned values. Several model forms are commonly used, and usually some subjective decision about what model to use is required. Semivariogram models usually include one or more parameters that can give useful information about the spatial correlation structure of the data, such as the maximum distance over which data values are correlated, or the variability of our data over very short spatial scales. Once our semivariogram model has been determined, we can now calculate our spatial weights and use our data to make predictions. The result is a grid of values showing estimated tap water hydrogen isotope ratios. Note that we can make predictions for areas far from samples, as well as areas over the ocean where tap water doesn't exist. Some common sense is required here, but because the semivariogram model represents the relatedness of values as a function of distance, Kriging methods can also provide direct estimates of prediction uncertainty. You can see here that uncertainty increases as the proximity of samples decreases, which should make sense if we consider the premise of spatial autocorrelation. It is important to recognize that the Kriging uncertainty is an incomplete estimate, in that it does not consider uncertainty in the form or parameter values of the semivariogram model. For our application, the prediction and uncertainty maps together provide a basis for identifying regions of the study area where the water that people consume falls within certain isotopic ranges. This information underlies the use of stable isotopes in migration ecology and forensic applications, which you can learn more about through other videos in our series.